So we're going to be continuing our discussion of macromolecules. And of course, there's two types of macromolecules that we are talking about. The first is the proteins, which we spent a lot of time on. And now I want to move on to the nucleic acids, which, as we'll see, are not as complicated in terms of their structure and function, so it won't take us as long. And of course, the nucleic acids are made up of polymers also, but these polymers are nucleotides, and we'll look at those in some detail. And of course, there's two types. There's DNA and RNA, and we'll look at the differences between those. And so the reading for these is marked at the bottom here, 173, 178, and 259 to 262. Okay, let's go to the next slide here. Okay, so nucleic acids, this is the macromolecule we're talking about now. They're polymers of nucleotides. And we'll look at the nucleotides in a few minutes. And again, there are two types. There's DNA and RNA. And let me start by just giving you a little bit of overview of DNA. DNA has one function in the cell, one function only, and that is it stores information, that is the genetic information. It has no biological activity per se, it carries out no reactions. It just sits there relatively inert, but it carries the instructions, of course, for the entire organism, so information storage. Now, we talk about the sizes of nucleic acids. Just like we talk about the size of proteins in the terms of the number of amino acids, we talk about the size of DNA in the number of nucleotides. And so the smallest naturally occurring DNA molecule that people have seen is probably around 3,000 nucleotides. Maybe a little bit smaller, but 3,000 is around the smallest size we see, and those usually occur in viruses, tiny small viruses that have small, usually circular genomes of 3,000 nucleotides. It can either be double-stranded or single-stranded, depending on the virus. So 3,000 is like the small limit, small end of the range. And then we have human chromosomes, which are much larger. You can have an average human chromosome around 100 million in length. The largest chromosome, I don't remember what the number is, but there's around 250 million. So these are incredibly long polymers, much longer than any protein. Proteins, we may have 27,000 or 30,000 max. Here we're talking about as many as 250 million. Could be even longer. Now, if we look at RNA, let me say this in this way: RNA also can carry genetic information. In principle, it's the same. It's the sequence of nucleotides that carries genetic information. So it can clearly carry genetic information. We know many viruses have RNA genomes. Coronavirus, COVID-19 has an RNA genome. Poliovirus, HIV. Other coronaviruses like the common cold virus, there's many viruses have RNA genomes. And in terms of the range, the smallest RNA molecule that we've seen that has some function is around 10 nucleotides. And when we look at DNA synthesis, we'll see where these fit in. But the smallest is around 10 nucleotides, and then the largest is at most a few 100,000 nucleotides. So an average messenger RNA, for example, is around 2,000 nucleotides. So these are much smaller than DNA, ranging between 10 and maybe 100, 200,000 nucleotides. Now, there's a huge difference between DNA and RNA. Although on the surface, and we'll look at the structures, they can look very similar. They have some very important differences, although they're tiny, but those tiny differences make an incredible change in the activity of the RNA molecule. So while DNA only stores genetic information, RNA has many functions. There are many types of RNA. Of course, we know about messenger RNA, we know about transfer RNA, we know about ribosomal RNA, but it turns out there are dozens and dozens and dozens of other types of RNA molecules in the cell. And they carry out many functions. And I have a short list here below. They, of course, can carry out information storage just like DNA. They can be involved in regulating gene expression. RNA molecules are involved in protein synthesis. And I'm not talking about transfer RNA, ribosomal RNA, and messenger RNA. I'm talking about other types of RNA molecules that can regulate the process. RNA molecules are involved in DNA replication. And RNA molecules can be enzymatic. I mentioned that when I talked about enzymes, that proteins can be enzymes, but also RNA molecules can be enzymes as well. So RNA has many, many more activities than DNA. And it has some implications really about the origin of how cells originated and what their kind of genetic basis was, but we'll talk about that towards a little bit later towards the end. So here's the subunit of nucleic acid. I like to use my drawing because there's something similar in the book, but this kind of illustrates some of the important points that I want to bring out and kind of elaborate on a little bit. So this is a nucleotide here, and the nucleotide basically contains 3 different sections. There is the nitrogen-containing ring, which is the base, and there's 5 different bases that are used in nucleic acids, and we'll look at those. There's the sugar in the center. It's a 5-carbon sugar, which is called a pentose. And then there's the phosphate that comes off of sugar. And so those are the 3 different elements in the nucleotide. So phosphate, sugar, and base. Now if you look below I have it drawn out more as the chemical structure. And so you can see the base there. That's one of the 5 bases. That's cytosine. And you can see it's a ring-like structure. Nitrogens are in there. Then you have the sugar. It's a ring structure, and there are 5 carbons, and they're numbered, which becomes important. So the number 1 carbon has the base attached to it. Number 2 down below. Number 3 you see an OH. Number 4 carbon in that corner there, and the 5th carbon is off the ring. And you can see that's CH2. Now, for those of you who haven't had any organic chemistry yet, the numbering, so if you have a diagram like that, a drawing, you don't have to draw in all the carbons and hydrogens every time, but the carbons are at the corners. And it's assumed that there are hydrogens coming off of each carbon unless something else is drawn, like the OH. So for the number 2 carbon, there's a carbon's going to have 4 bonds, and so it's attached to the neighboring carbons, and then there's 2 hydrogens coming off of that. And yeah, so we always draw them as these little angles that represents a carbon. And then you see the phosphate. That's a triphosphate. This coming off of the CH2, and this is basically the same kind of triphosphate that's used for ATP. ATP is very similar, except it has a different base and the sugar is slightly different. But those are 3 high energy phosphate bonds, and they have different names. The one closest to the carbon is alpha, the one in the middle is beta, and the one at the very end is a gamma phosphate. Now in terms of the nomenclature, so we have the the bases, which we'll look at in a little bit. When you have a base like cytosine just attached to the sugar with no phosphates, it's referred to as a nucleoside. And it says deoxynucleoside because this is a subunit of DNA. And then the minute you add a phosphate, it could be a single phosphate, which would be called a monophosphate, or two phosphates, which would be a diphosphate, or the third phosphate, which of course would be the triphosphate. Anytime there's a phosphate on there, it's referred to as a nucleotide, not a nucleoside. So we have deoxycytosine, cytidine monophosphate, abbreviated as DCMP. Then DCTP, for the diphosphate, and DCTP for the triphosphate, and the D stands for the DNA, or deoxy sugar, which we'll see in a second. So that's the kind of nomenclature around the nucleotide. Now let's look at the sugars. So here are the two sugars that are used in nucleic acids. It's just what I showed you. Let's look at the DNA on the right. This is the sugar that's used in DNA. Again, 5 carbons, and this one's not attached to a base. And you see the number 1 carbon has the OH coming at the top. The number 2 carbon has the red hydrogen. The number 3 carbon has OH. Number 4 is that corner on the left, and then the CH2OH is the number 5 carbon. So this is a deoxyribose. I'll come back to that in a second, used in DNA. If you look at the sugar that's in RNA, quite similar. Looks basically the same. The only difference is that on the number 2 carbon, there's an OH. So this is ribose. And it turns out that number 2, that we call it 2'. So 1', 2', 3'. The 2' hydroxyl, which is that OH, completely changes the properties of the nucleic acid. It makes it reactive, and we'll look at that a little bit. The DNA that's used, the deoxyribose that's used in DNA is missing that 2' hydroxyl, and that's why it's referred to as deoxyribose. Actually, it should say 2-deoxyribose, because it's deoxygenated at the number 2 carbon. So 2-deoxyribose. So these are the two sugars that are used in RNA and DNA, ribose and deoxyribose. Let's quickly look at the different bases. And there's not a lot I want to say about them, but I just want you to be able to look at them for a little bit with some patience, in a sense. So these are the nitrogen-containing bases that are present in both RNA and DNA. And first off, there are 2 classes. There are the pyrimidines. These are the ones shown on the top. The pyrimidines are a single ring structure. 6 atoms in that ring. Some of those atoms are carbon, which are not shown. It's just that angle. And then nitrogens. So these are 6-ring compounds, one single ring. Those are the pyrimidines. One of the things I want you to look at is that they look pretty similar. There's some differences, but they look pretty similar. The dimensions are very similar. Now, on the bottom are the purines, guanine and adenine. And the difference there is that there's a double ring structure. So these contain nine atoms in those rings. So the purines are larger than the pyrimidines, and that's kind of the way I remember them. It's like, the purines are larger. The shorter name is the larger compound. The pyrimidines, which is the larger name, is the smaller compound. It's just a kind of a trick for me to remember which are which. So pyrimidines, cytosine, uracil, and thymine. The purines, guanine, and adenine. And again, look at the guanine and adenine. They kind of look similar. Now, ultimately, when they undergo the hydrogen bonding, which we'll see when you make a double-stranded molecule, the regions that interact are on the right-hand side, towards the top. So with the cytosine, let's talk about the uracil. So the uracil has the double bonded O, NH, and then the double bonded O. And if you look at the thymine, similar thing, double bonded O, NH, double bonded O. Those are the regions that interact during the base pairing process. So in RNA, let me start like this. In DNA, the two pyrimidines are cytosine and thymine. In RNA, there is no thymine, it's uracil instead. So RNA has uracil and cytosine, and guanine and adenine. And DNA has cytosine and thymine, and guanine and adenine. So the only difference there is that the uracil in RNA and thymine in DNA. But what I want you to look at in terms of the genetic information and the base pairing ability, uracil and thymine are identical. They're identical. The only difference with the thymine is that CH3 coming off on the left. Otherwise, they're identical. They serve the same function in terms of the genetic code and in terms of base pairing. So they're very similar. And then, of course, they're abbreviated: C for cytosine, U for uracil, T for thymine, G for guanine, A for adenine. Again, I know you've had a lot of this in introductory biology, and maybe you've had genetics. But I just wanted you to take some time. I kind of like to present the way I see these things. And so just spending a few moments to look at how similar these are, I think, is important. So we looked at sugars, the difference between DNA and RNA, ribose to deoxyribose, and we looked at the bases, and again, the differences between DNA and RNA. Now these nucleotides link together. And I'm going to show you again one of my drawings, but this in the next one shows you how they link together. So I hope you can see that okay. So, of course, what it says is the linkage, which is actually the polymerization, occurs through the 3' hydroxyl of one nucleotide. So let's look at the DCTP in the top. Again, the numbering, 1, 2, 3, that's 3' hydroxyl. And the 5' phosphate of the second nucleotide. So below you have DATP. We know this is a DNA molecule, or it will be a DNA molecule once it's polymerized, because there's no oxygen coming off that number 2 carbon. And so what happens is, during the polymerization process, the oxygen, which is the number 3 oxygen, links to the phosphates, the alpha phosphate on the one below. And then it releases that diphosphate, those 2 phosphates. And so you end up getting a new linkage, which is referred to as a 3' to 5' phosphodiester bond. 3' to 5', indicating that 5' carbon, phosphodiester bond. Now, before I move on to show you what it looks like when it's polymerized, first of all, of course, the polymerization takes place during DNA synthesis using DNA polymerase. But what happens is, just like with the proteins, where you have the new amino acid linked to the carboxy end of the molecule, here it's similar in a sense that the next nucleotide that would be added, let's say there was a DTTP that was going to come in next, it would add on to that last 3' hydroxyl on the DATP. So that free 3' hydroxyl is where it's added onto. So often you hear about the terms, like the synthesis, is 5' to 3'. 5' up at the top, 3' at the bottom, and then it's always added to the 3' hydroxyl. So let's take a look at what these phosphodiester bonds look like. So on the left we have basically showing a dinucleotide, but it looks like it's much longer. They're only showing 2 nucleotides, and they're showing just a random base. They're not showing which base it is, but it's a pyrimidine linked to the sugar. And so the sugars that I want to focus on, of course, they're in blue. And so you have the phosphodiester bond, which is linked between the 3' hydroxyl, and you get the phosphate, another oxygen, linked to the 5' carbon. And the orientation is such that you have what we call the 5' end of the chain and the 3' end of the chain. The 3' end of the chain is basically at the end, it would be a free 3' OH. And at the 5' end of the chain, if you were to go all the way to the end, the very first nucleotide in that chain would have a triphosphate. Because you have the first triphosphate, and then the next nucleotide is linked onto the hydroxyl. So a couple things that I just want to say about this. Often you hear that the nucleotides are comprised of what's called a sugar phosphate backbone. And that's because if, let's say, you have a long length of this, let's say you have, 300, 500, 1,000, 1,000,000, it's basically a linkage of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, as long as you go, phosphate, sugar, so phosphate, sugar, backbone, or sugar, phosphate, backbone. And then the bases are coming off of the number 1 carbon, and of course those bases vary depending on the sequence. And that variability in the bases is what gives the genetic information. So you have sugar phosphate backbone. The other thing is that it's a highly polar, hydrophilic. Why is that? Lots of oxygens and charges. And it's also referred to as an acid. Its DNA is deoxyribonucleic acid. It's an acid because at neutral pH it gives off the hydrogens that otherwise would be attached to that oxygen, which is negatively charged in that yellow triangle. So sugar phosphate backbone, highly polar or hydrophilic, and slightly acidic. So now let's just move over to the other side. On the right-hand side, again, same thing. Sugar phosphate, sugar phosphate. You can see the phosphodiester bonds. Here they're showing a stretch of 4 nucleotides. And this is a sequence or a structure of RNA. How do we know it's RNA? First of all, we know it's RNA because it has a 2' hydroxyl. Second of all, we know it's an RNA because it has uracil. If it was DNA, it would be thymidine or thymine. Now, you can have RNA sequences that don't have any uracils. So it could have been CA, GGCC, whatever, with no uracils, but we would still know it's RNA because of that 2' hydroxyl. Now, the 2' hydroxyl, which is the only difference between the DNA and the RNA, that 2' hydroxyl completely changes the chemistry of the RNA molecule. That 2' hydroxyl is reactive. It can carry out reactions. And let me just show you, whereas DNA cannot carry out anything, it's pretty inert, it just sits there. So one of the simpler reactions is what's called autohydrolysis. So let me just give you an example of that autohydrolysis in the next slide here. So here it says RNA is reactive and can undergo autohydrolysis, self-cleavage or self-cutting. So what's shown here is a stretch of RNA, an RNA chain, and it's showing only 2 of the sugars, look on the left-hand side, it's showing sugar, but then there's, if you look carefully, there's phosphate, another base, phosphate, another base, phosphate, and then the sugar that's being shown. And what happens is if you have a long stretch of RNA, then with that spacing, that 2' hydroxyl, the molecule can bend and kind of loop around. So that 2' hydroxyl can react with the phosphate below. And there's that line with the arrow. Can react with the phosphate below and cleave that phosphodiester bond and make a new linkage where you have the phosphate attached to the 2' hydroxyl. It makes kind of like a circular molecule. So the RNA can cut itself. Now, what this shows is that there's a reactivity to the RNA. It can cut itself. It can cleave itself. It's a real activity, and that's the first clue that maybe RNA can do things in the cell that DNA can't do. So even though this self-cleavage autohydrolysis looks relatively simple, it is the basis for all the other things that RNA can do enzymatically in the cell. Now, DNA, on the other hand, doesn't do that. So you can take DNA, you can put it into a test tube, and you can let that test tube sit for days, weeks, months, years, 1,000 years. In fact, they've recovered DNA from prehistoric mammoths and cavemen teeth and that kind of stuff. So the DNA is stable, can sit there forever, doesn't change. That's in part why it's a very good genetic storage material, because it's very stable. On the other hand, if you take RNA and put it into a test tube in water and put it on the lab bench, by the time you come back overnight, most of it will be gone. It'll be chewed up. It chews itself up, carries out this autohydrolysis. Of course, if you raise the temperature to 37 degrees, maybe even a little bit higher, it'll disappear in a few hours. Okay, so that's the ability to undergo self-cleavage. So RNA has this biological activity, and we see that in a particular type of a molecule, which is called a ribozyme, which is an RNA-based enzyme. So this illustrates simply what a ribozyme is. It's a biologically reactive RNA molecule. Due to the reactivity of RNA, some RNA molecules function as enzymes, and this is an example. We know that RNA can cut itself. We saw that with the autohydrolysis. But here in these are specific RNA molecules, ribozymes, which cut other RNA molecules. So it shows on the left, the ribozyme has this kind of structure with these two globs coming off, but it can base pair because of its base pairing ability. It can base pair with other RNA molecules like a messenger RNA, and because of the reactivity of the 2' hydroxyl, it can cut that RNA molecule. So these ribozymes, this class of ribozymes cuts other RNA molecules, so it acts as an enzyme. And in fact, splicing, which we'll talk about much later in the semester, splicing in part occurs through the ability of the RNA to cut itself. So RNA is clearly reactive. It can act as a ribozyme and cut other RNA molecules. And people have actually tried to construct ribozymes that would be specific for specific viral RNAs, like HIV. That is, then you would introduce this ribozyme into the cell, and it would specifically cut the HIV RNA. or coronavirus RNA. And people are working on that, and there's been some success, but obviously not anything that can be used therapeutically. So RNA is reactive, and it turns out there are many, many types, as I mentioned at the beginning, many types of RNAs in the cell. There's messenger RNA, there's ribosomal RNA, there's transfer RNA. We've heard tons about these. There's ribozyme, which is enzymatic, can cut other RNA molecules. RNA primers, which are used in DNA replication. There's what are called microRNAs, which are small RNA molecules that can regulate gene expression. There's small nuclear RNAs involved in mRNA processing or splicing. There's long non-coding RNAs, regulate transcription, gene expression, chromosome behavior, and there's what are called NAT RNAs. These are naturally antisense occurring molecules that have complementary sequences to other RNA molecules, and they can bind and cause changes in their activity. So there's many, many types of RNAs in the cell, and there have been many different types of RNA-based enzymes that have been identified aside from the ribozymes. Let me show you. I have a table here. So this table shows basically some of the types of RNAs that have been shown to have enzymatic activity. So we talked about the ribozyme. It can cut other RNAs. Well, it has ribozymes. Actually, on the right-hand side, it's using ribozymes as a general term for an RNA-based enzyme. But traditionally, ribozymes refer to the RNA, the original ones that were found to cut other RNA molecules. But if we use this table, there's a whole list of other RNA-based enzymes. So ribosomal RNA, which is one of the components of the ribosome, actually is the enzyme that carries out the peptide bond formation and protein synthesis. That protein bond, that generation of the peptide bond, is not carried out by a protein enzyme, but it's an RNA enzyme. Then I mentioned splicing, that there are small nuclear RNAs that are involved in splicing and self-splicing, so this is very important. And then all you have below, what it says in vitro selected, means that it hasn't, that these activities, the DNA ligation, RNA polymerization, RNA phosphorylation, and all these other activities, these have not been seen in cells, but they've been generated in the lab, meaning they've been constructed in order, a specific RNA sequence has been constructed to carry out DNA ligation. DNA ligation is the joining of two DNA molecules, or they've been selected or constructed to carry out RNA polymerization, meaning it can actually function as an RNA polymerase and make an RNA molecule. So the finding that there are many, many, many different types of RNAs in the cell, and that many RNA molecules have these enzymatic activities, has led people to think about what was in the origin of life, what was the original genetic material? DNA, or RNA, or does protein come first? I mean, how did things get started? So people have kind of come to the conclusion that it must have been RNA. That at one point, the primitive cell, was based solely on RNA. So why is that? One is that if you have a DNA genome, you have to have a way to make the DNA, to polymerize it, to synthesize the nucleotides. You have to have a way to do that. And DNA doesn't have any activity, so it's not likely. Proteins probably couldn't have come first because, again, there's two things. One, you need to have the information to generate the protein, the genetic information. And the second is that how are these proteins made? They can't make themselves. And then, so people realized that probably it was RNA, and that RNA formed the basis of life at the very beginning, because RNA can do all these things. RNA can be genetic information because of the nucleotide sequence, and it can carry out enzymatic activity, so it can make itself. So we see here one of the possibilities is RNA polymerization. So it can make itself. It can also make proteins, because it can bind peptide. It can bind amino acids together. So the thought is that the earliest forms of life on Earth were probably based on RNA. And people have referred to this as the RNA world. That is, RNA was the essential genetic and biochemical information in the cell. And so this was a book that was published by three individuals. The individual in the middle, Tom Cech, I actually know him. I worked with him for a while in Colorado. But Tom Cech was actually the first one to ever discover a ribosome. So he was the original discoverer of a RNA active enzyme. And he ended up getting a Nobel Prize. So what it says, the RNA world, the nature of modern RNA suggests a prebiotic RNA world. And it's interesting, even if you look at, well, let me back up a second. So the RNA world came first. And since RNA is inherently unstable, then the thought is that what happened was that the molecules began losing that 2' hydroxyl to become stable and to become DNA. So it can work, but you can't, it's not genetically stable, it's not biochemically stable, so the loss of the 2' hydroxyl made the DNA molecule much more stable information storage, and it's also thought that thymine, which is present in DNA, is more stable chemically than uracil. And it's interesting for a lot of, there's a lot of evidence that kind of points to this, like ATP, which is used as an energy source, for the cell, is a ribonucleotide. And if you look at the synthesis of the deoxynucleotides, they start out as ribonucleotides, and then they eventually get converted into deoxyribonucleotides. So that even the pathways, RNA kind of comes first. Anyhow, so this the next slide kind of sums this up. That RNA may have preceded DNA as proteins in evolution. Of course, we have the RNA-based systems, which we understand now. Then it says evolution of RNA that can direct protein synthesis. And so we know that RNA can self-replicate, and it can also make protein. And then finally, DNA takes over as genetic information because DNA is more stable with the loss of the 2' hydroxyl. And then RNA becomes the intermediate between the DNA and the protein in the ultimate synthesis of proteins on the ribosome. Okay, so that's an introduction, and then we're going to start talking about double-stranded DNA and what some of its properties are. And this is where we're going to end this one here.